Lecture 1. Amino acids. Functions of amino acids. A variety of roles in metabolism. The building blocks of proteins. Forming parts of coenzymes. As precursors for the biosynthesis of molecules such as hem. Standard and non-standard amino acids. More than 300 different amino acids have been described in nature. Standard amino acids. Only 20 are commonly found as constituents of proteins. Non-standard amino acids. Amino acid derivatives found in protein. Non-protein amino acid. Why are amino acids uniquely suited to their role as the building blocks of proteins? 1. General structure of amino acids, very important. Carboxyl group, amino group, side chain, or R group, R equals remainder of the molecule. 2. Stereochemistry of amino acids. Configuration. For all the common amino acids except glycine, the alpha carbon is bonded to 4 different groups. The alpha carbon atom is thus a chiral center. Chiral, from Greek chir, meaning hand. An object or a system cannot be superimposed on its mirror image. One hand does not match the other when superimposed. Chiral molecule and enantiomers. Chiral molecule. If a molecule has an atom bonded to 4 different groups, it can be chiral. Enantiomers. A chiral molecule has left and right-handed isomers, called enantiomers. The 2 enantiomers of each amino acid are designated by D, L system according to the D and L glyceraldehyde. D, dextrorotation, L, levodopa. Only the L amino acids have been found in proteins. D isomers have been found only in small peptides of bacteria cell walls or in some peptide antibiotics. 3. The classification of 20 standard amino acids, very important. The name and abbreviation of amino acids. All the AAs were given a trivial, common, name. Glutamate from wheat, gluten. Tyrosine from cheese, tyrosine Greek. Each AA is given a 3-letter abbreviation and 1-letter symbol. They often the first 3-letter and the first letter. When there is confusion, an alternative is used. They should be remembered. Classification of amino acids. Based on polarity. Nonpolar, aliphatic amino acids. Polar, uncharged amino acids. Aromatic amino acids. Acidic amino acids. Basic amino acids. 3.1 Nonpolar, aliphatic amino acids. Gly Alabama, Pro, Val, Lu, and Eel. 5-membered ring structure, rigid in conformation. Gly. R group, hydrogen. Symmetric, not chiral. Pro. Amino acid. 5-membered ring structure, rigid in conformation. Alabama, Val, Lu, and Eel. Hydrocarbon R groups, often involved in hydrophobic interactions for stabilizing protein structure. 3.2 Polar, uncharged amino acids Air, THR, cis, SN, GLN, and MET. R group hydrophilic. Hydroxyl groups. Sulfur atoms. Amide groups. Disulfide bonds. SH group of 2 cis in proteins can be oxidized to form a covalent disulfide bond. Disulfide bonds play a special role in the structures of many proteins by forming covalent links. Disulfide bond. 3.3 Aromatic amino acids Phe, TIA, and TRP. Phe and TIA, benzyne rings. Tryptophan, indole ring. The O group in TIA is an important functional group in proteins. Phosphorylation, hydrogen bond, etc., polar. Proteins in solution absorb UV light with absorbance maximum at 280 nanometers. They are jointly responsible for the light absorption of proteins at 280 nanometers. Proteins in solution absorb UV light with absorbance maximum at 280 nanometers. Measuring protein content by photospectrometry. 3.4 Acidic amino acids. Aspen glue. Have carboxyl in their R groups. 3.5 Basic amino acids Lis, Arg, and his. R groups aminoguanidino. Imidazole. Positive charged R groups at pH 7.0. Note these structural features. 1. All 20 R amino acids. 2. For 19 of the 20, the amino group is primary. For proline, it is secondary amino acid. 3. Except glycine, the carbons for 19 of them are asymmetric, or chiral. Polar, uncharged amino acids. Non-polar amino acids. Alabama valulyl glyprofetrp. Polar, uncharged amino acids. Aromatic. SRTHRCIS asngylin metia. Acidic amino acids. Asp glue. Basic amino acids. Lis Arg his. 3.6 Non-standard amino acids. Amino acid derivatives found in proteins. 4-hydroxyproline and 5-hydroxylysine in collagen. 6-Newton-methyllysine in myosin. Many additional non-standard amino acids are found in cells, but not in proteins. Ornithine and citrulline. Intermediates in amino acid metabolism. 3.7 Essential and non-essential amino acids. Essential amino acids, or indispensable amino acids, cannot be synthesized by the humans, must be supplied in the diet. 8. Phi, Val, THR, TRP, EL, Met, Lu, Lis. Semi-essential amino acids. 2. His and Arg. Required by infants and growing children. 4. Acid/base properties of As, very important. Amino acid has both a basic amine group and an acidic carboxylic acid group. In neutral solution, pH 7.0, the amino acid contains a negative charge and a positive charge. It is called as Zwitterion, German for hybridine. A plus A0 A cations Zwitterion anion. A is ionized to various states depending on pH values. Pi, there is a specific pH, designated isoelectric point, pi, at which an AA has equal positive and negative charge, no net electric charge. Very important. Plus H plus. R, CHQ. NH3 plus. R, CHC.O.O. NH2. H plus. pH pi. pH equals pi. pH pi. A plus. A0. A. Cation. Zwitterion. Anion. Five. Polypeptides peptide bond. Peptide bond, the special name given to the amide bond between the carboxyl group of one amino acid and the amino group of another. Amino acid residue. Peptide bond. Characteristics of the peptide bond. The peptide bond have partial double bond feature. About 0.132 nanometers, C single bond, 0.149 nanometers, C equals and double bond, 0.127 nanometers. Rigid and unable to rotate freely. C H N C O 0.153 nanometers, 0.146 nanometers, 0.132 nanometers. The peptide bond is planar, transconfiguration and uncharged. Peptide plane, the 6 atoms attached to the peptide bond are coplanar, the carbonyl oxygen and the amide hydrogen are in transpositions. The peptide chain is directional. Amino terminal or N-terminal, the end having a free A amino group. Carboxyl terminal or C-terminal, the end having a free A carboxyl group. By convention, the N-terminal is taken as the beginning of the peptide chain, and put at the left, C-terminal at the right, polymers of amino acids. Peptides can be classified according to how many amino acids they contain. Dipeptide, 2 amino acid residues, tripeptide, 3 residues, and so on. Oligopeptide, 12-20 residues. Polypeptide, many residues. Biologically important peptide oxidized form glutathione. Glutathione, GSH. Tripeptide, glutamic acid, cysteine and glycine. Function, important in biological oxidation reduction reactions, has reduced and oxidized form. It's the most important molecule you need to stay healthy and prevent disease. Glycine. Cysteine. Oxidized form glutathione. Disulfide, GSSG. Reduced form. GSH. Glutamic acid.